this lab is the Diffusion Osmosis lab. And we'll use the gelatin tube to demonstrate diffusion, and we use what's called dialysis tubing, which is that plastic bag, right? Dialysis tubing, it's open on both ends, so you have to use these clamps. So you clamp one end, added a solution into it. I'll explain what that is in a minute, and I'll explain what the bag is too. Okay, it's considered a semi-permeable membrane for our purposes. So diffusion is what happens when something goes from, say, high concentration to low concentration. The only reason it's diffusing is because there's a difference in concentrations, right? And so once, let's say it's equal, then you'd say the diffusion is stopped. So it's basically movement of something from high to low. Now it can be inside this room, like say the air. It could be in a solution. We're going to use the gelatin tube to demonstrate it. So we have a tube or tubes, right? And in the tube is water. And there's also gelatin, which is a protein, right? So the protein would be very small molecules. All right, and it's when you use gelatin to make Jell-O. So you put about 5%, so much protein in there, and that just sort of makes it solidifies it. Now, if we didn't have that in there and we wanted to do diffusion, we could just add a drop of a dye to water, and you would see over time that the drop of dye diffused, and it would diffuse in the water until the water was all one color. Right, that's an example of diffusion. And it stops diffusing when it's equal concentration in whatever water container it's in. So we'll put gelatin in there to just slow it down some, so it doesn't occur so quickly. But unfortunately, it tends to be, kind of slow as well. When we add the dye, then we'll say the dye is going to start at the top of the tube. So I've added a blue dye to the top of this gelatin tube, and then we expect the dye to move into the gelatin or diffuse down. Now, if we let this go for maybe a month, it might take a long time, the whole thing would turn blue. If it was equal, then it would appear blue, the same blue color all the way through. But that would take a lot of time. And this experiment, for this to really move any measurable amount, it takes a couple hours. Now, let's see, do I have one from this morning? So this morning I added blue dye to the top of this tube, and now it's about a millimeter or two down from the top, but not too far down. I've actually added more dye than I needed to this. So if I took off, let's see if I can do that. So I take off the dye, it doesn't look like there's so much in there now. But that's, it's moved a little bit down compared to this one. It's still at the top of the gel, but it's kind of hard to tell. Now the yellow dye that I've added is going to move a bit more quickly than the blue dye, and it's already gone into the gel a little bit. So we're saying that these dyes are diffusing in that direction from the top of the gel. Now there's a couple things that affect the rate of diffusion. The rate means like how far does the dye or how far does something diffuse in an hour or a minute or something like that. So the molecular weight influences how quickly these dyes move. So when something diffuses, it takes time, I say, and it also takes some temperature, right? Everything, it doesn't look, when you look at this gelatin tube, it looks pretty solid, and you would say, well, nothing's moving in there, right? I mean, just by looking at it. But just like everything is either vibrating, the molecules are vibrating, and sometimes they're moving up and down, and right, they're actually moving from a location, but not much. Now these air molecules, they're moving and they're vibrating when we breathe in the air. That's why things diffuse, right? Because when they get into when the molecule gets in the air, things are hitting it, bombarding it, causing it to keep spreading out. If the molecules weren't moving, there wouldn't be diffusion, right? That's basically what happens. You take a glass of water like this, it doesn't look like, if you hold it still, you don't think, well, none of the water molecules in there are moving, right, if it's still. But they are, right? They're just not moving a lot, but there's movement. Even when you freeze water, it turns into a solid. The molecules in the ice are vibrating. So it has a temperature. So what is molecular weight? It tells you how big a molecule is. So let's say the yellow dye is around 200 and something, I don't know, it might be 200, it might be closer to 300, but let's just use 200 as a nice easy number. I think that's why that's there. Compared to the blue dye, it's bigger, it's around 700, right? So the molecular weight of the blue dye is a bit more than the molecular weight for the yellow. So what would you expect? Would you expect this one to diffuse faster than this one, or vice versa?
The smaller one, the yellow.
Right? You would think, and that's the case, right? Because it's smaller, it can move faster, right? There's nothing so impeding it as much. It's just like if you're playing football, the little guys normally move quicker than the big guys. Same kind of scenario. Kind of that's what's happening. So when you increase molecular weight, the rate of diffusion, it's going to diffuse more slowly. So that's one of the parameters. I don't think I've used parameter before. One of the parameters that we would do in this experiment, if you were doing these on your lab, I would tell you to add drops of the dye to the top of these tubes, and then in an hour, we would measure how far the dye went down into the gel. Right? We would take a measurement in millimeters. So we'd say if it starts there, and then it ends, it keeps moving, say down that far, then we'd measure the millimeters that it went down or diffused. It's sort of a crude way of measuring diffusion. Probably another way, better way of putting it. Now what do you think? What would temperature affect? If we put these tubes for an hour and left them on ice and added the dye, and then compare the ones on ice to the ones that are at room temp. What do you think?
It'd slow them down.
Yeah, right, because it's colder. The colder the temperature, the slower things are moving. Right, just like in this room, if it's 7 degrees in this room, they're moving slower the molecules than if it was at 80 degrees. So by increasing the speed of the molecules of the dye and the molecules of water, that would, right, speed up the rate of diffusion. So those are the two of the kind of concepts that you want to learn. You know, it's a good idea to have a rough idea of what diffusion is, right? That things tend to want to go from high concentration to low. If you want to go from low to high or concentrate something, then you need to put work or energy into the system, whatever that might be. You know, I think in class they said if you want to put the perfume back in the bottle. Did I use that example? Right. I think that makes it memorable. Because you can't do that, right? It doesn't make any sense. But if you open up a bottle of perfume, now the only reason you know the perfume is diffusing is because of the smell. So what I have here is a dialysis bag, and in the bag, or dialysis tubing, you could call it that too, there's what 30% sucrose has been added to it. And then the bag, I'm going to put, after I weigh it, it weighs 36 grams. This morning it went up by a gram. And I'm going to put it in some water. And then I'll reweigh it and see what happens, okay? So what, in this experiment for osmosis, right? Osmosis is the diffusion of water across a semi or selectively permeable membrane. Now membranes in the classic sense are found around cells or vesicles or the organelles, and it's semi-permeable as well. But instead of, in this experiment, the dialysis bag or the dialysis tubing is considered semi-permeable. Why is that? It's because it has holes in it and the holes will allow water in and out of the bag, right? So we've got the dialysis bag now in this flask, which contains just water. So if you think about it, let's say this is a line, this is actually in your textbook, and we'll represent this side as being outside the bag, and then on this side we're talking about 30% glucose or sucrose inside the bag. And we have water molecules, let's say they're like this circle here on both sides, right? There's lots and lots of water, but they can go through the hole. They're small enough. But in this side, we've got sucrose. Right? We'll draw them as like rectangles. And the sucrose can't fit through, the holes are too small for the sucrose. So they have to stay, the sucrose has to stay in the bag. So what we want to do to demonstrate osmosis, right, which means water molecules are moving across in and out of the dialysis tubing. So what do you think? Will more water molecules go this way into the bag, or will more water molecules go out of the bag?
Well, depending what is higher and what is lower concentration. So-
Yeah, you know, there's no, I used to always think, well, you wanna dilute the 30% out. That's how I used to remember it. And that's what happens, right? Because more water molecules is gonna go into the bag. Because, and your textbook explains it, that the sucrose blocks the holes. It gets in the way, it keeps the water from going. They still go that way, but they're more likely to go in that direction than this direction. So that's one explanation. So we'd expect in an hour that this bag would weigh more because more water is going to tend to want to go into it. Now the problem with this experiment was when you pull out this bag, right, and it drips with water, right? So a lot of this water that's attached wasn't attached before we put it in there. Kind of screws things up a little sometimes. So you have to be careful, like, to get rid of most of the water. But that's the purpose, like the dialysis tubing is to demonstrate osmosis, which is the movement of water across the semi-permeable membrane. It's basically diffusion of water. So both cases, these are diffusion, and they would both be considered passive when you compare it, you know, to say something that's happening in a cell or a biological system. So when we apply the term passive transport, then we're saying that, right, no energy is required, and we're moving from a high concentration to a lower concentration. If it's active transport, then it's the opposite. We're trying to get the perfume back into the bottle. Then it's active transport, or we're concentrating, and we need a source of energy like ATP. Okay, and then if it's a biological system, and you have a membrane, right, say like that's the bilayer of a membrane, then you have proteins, transmembrane proteins, and then there might be a hole in the protein generated so that you have a hole now in the membrane from the protein. Now these proteins or transporters are selective in what they allow through that hole. So the aquaporin was an example that would allow water across the membrane through that pore. But that would be a, you know, say a passive transport. It'd be some sort of osmotic event allowing that. It's not. Now there might be some transporters that require energy, but I'm pretty sure the aquaporin example in class wasn't it was considered passive. But it's basically the so I think it's too early to weigh that bag right now, and it's starting to get, it's already moved about 2 or 3 millimeters down for the yellow. Now this morning's one has moved probably about 10 to 12. And this is like 4 hours. So the gelatin is a little too slow for a lab unless the lab lasted for 2 hours. Right, we started the lab this morning at 9, so I think I added that at 9, so it's actually been 5.5 hours. Devices, so, right, this is what we went over today. The gelatin tube was used to measure the rate of diffusion, is another way of saying it. How quickly things diffuse. If the molecular weight, the higher the molecular weight, then the slower you'd expect the diffusion to occur using these tubes. And if you increase temperature, you'd expect it to go up. Pretty straightforward. And then the dialysis, I put tubing here. Right, so I might ask you, what was the name of the membrane, or, I think one of the questions on the practical is, what, the dialysis tubing is considered what type of membrane, right? Would be semi-permeable. What passes through it? Why doesn't sucrose pass through it? You know, those kinds of things.